Hello and welcome. I'm Aaron Haubner from Celartia Incorporated. We're a Columbus, Ohio-based company that manufactures specialty bioprocess and cell culture ware. And today I'm sharing some of the methods and results that we got using our flagship product, the Petaka G3, in a thin film cryopreservation experiment. So I want to start by reviewing the limitations and drawbacks of traditional cryofreezing, particularly cryofreezing in vials. There's significant limitations, particularly when you're talking about adherent cells, because you have to lift them up off of the plate and transfer them into the vials. And then, of course, because you can't do thin film freezing, there's injury from, you can only do the slow freezing and slow thawing methods, which have to be managed, and there can be some damage from that. Plus, you suffer from some high post-thaw DMSO. You have to centrifuge or wash the cells often, and of course, you can't retain the original extracellular matrix or the original tissue structure. There's a long wake-up period when you recultivate the cells. They have to attach, and there are inherent limitations of the cryovial with regard to easy reading and unique device identification that we're all familiar with. So things can be improved. The problem with the cryovials is inherent in the design, really. It suffers, its major setback is it suffers from a very low surface area to volume ratio. Of course, this makes rapid freezing and vitrification practically impossible. Compare that to the thin film in the cartoon to the left, where you see a monolayer. And if you remove all of the excess liquid, you can achieve very rapid heat transfer. So somewhere on the order of 2 or 3 orders of magnitude. So it's the preferred thin film freezing for sheets or monolayers is preferred, obviously, because you can retain the intact structure. And people are working on this, so I want to do a little review of the literature here. Most are going to be familiar with at least one of these types of experiments. But Okawara vitrified skeletal myoblast sheets. They produced these, say, 1 centimeter approximately sheets, and they transferred them by lifting them off and attaching them to a polypropylene mesh, put them in these little plastic, looks like little plastic baggies, and then they submerge them in liquid nitrogen. And the method works. They get really good viability, very low apoptosis, and histologically they showed they preserved really well the structure of these sheets. So those are the benefits. Obviously, the drawbacks are it requires a harvest procedure. You're adding an extra matrix to it that may not be desirable in the final clinical application, and there's a complex transfer and manual cryopreservation process. So the proof of principle from the cell biology and cryobiology standpoint may be very good, but the practical application is challenging, to say the least. Similarly, Oliva vitrified oral mucosal epithelial sheets, and they played around in this study, played around with the vitrification solutions and protocols. They overcame some problems of tissue fracture, and then they lost a critical cell signal, and they optimized to retrieve that. So it's a good study, and they achieved all of their aims. But again, you can see some of the results here, some of the setup here, the sheets in a transwell put into this container that seems to be not fit for purpose. And there's a lot of open transfers involved. Again, if you're thinking about employing this benefit of flash freezing or maintaining cell sheets or monolayers for a translational study or a clinical study or even clinical applications, let's say it needs to be optimized further to really get the benefits from this method. The closest thing I could come to finding in the literature a custom-made device, a container for rapid vitrification or freezing, would be this device that was presented by Kindle et al., where they used a so-called twist method to vitrify PSCs. And there's no picture of it, but the device, the cartoon here, the drawing looks like basically a petri dish with a false bottom, which is kind of nice because you can do your cell culture on the top, and when you're done, you remove the media, get a thin film, and then flip it over, and you simply pour the liquid nitrogen into the false bottom, and you get direct contact with the underlying surface, and you vitrify very rapidly. And again, like the other studies, they compared that to, they got very good outcomes. They compared it to slow rate freezing in tubes, and all their markers and all their outcomes were very good, high viability, but they maintained their colony morphology, which was important. But even this being a custom-made product, there's a lot of openness. The lids are open, and I don't even know if this product is commercially available. So I would say it's maybe not necessarily GMP ready, let's say. So we were thinking this is an excellent method. There's a lot of advantages to thin film cryopreservation. So what we want to do is try to elevate the practicality of it and make it something more practical and scalable for the everyday researcher, or possibly something that could plug into a clinical trial. And that ideal process is really going to entail a few things: eliminate trypsinization in all transfers, minimize DMSO. If you eliminate the transfers, you don't lift the cells off the primary culture vessel, you're not going to have the thaw time, you're not going to thaw it, you're not going to have the long wake-up period and reattachment period. And ideally, you're going to do the whole process, culture, freezing, thawing, reculture, all in the same container so your product never leaves the sterile field. And ideally, that container is going to be already used in many GMP cell productions around the world. So that gets us to our product, the Petaka G3. As you can see, it's a very slim cell culture cassette, has 150 square centimeters of attachment surface, and fills to 25 ml volume. It has all the standard quality features that you would expect from cell culture ware. It's a little bit different. You're going to see it's very, very slim, so you can culture your cells in very high density comparable to the biggest multi-level flasks available on the market. I'm going to show you how all the liquid transfers are closed, so it's a functionally closed system. And you can do things like transfect, you can centrifuge directly, and the core technology actually grows your cells at physiologic oxygen levels and maintains CO2 even outside the incubator, so you have a lot of built-in advantages just on the basic culture. But we're going to focus the rest of this talk just on the cryo application. So the liquid handling with the Petaka G3 was achieved very easily. So for all of these experiments, safe blunt cannulas with syringes were preferably used, but for the addition of growth factors and other supplements, you can use multi-pipetters, repeater pipettes, micro pipetters, or serologicals if you have the tips. Basically, if it's a small standard tip in the lab, you can operate the Petaka through the sterile silicone port. Here's a picture of it in your standard benchtop workflow. I say if you can use a flask, you can use a Petaka. It's very simple. And here's our methodology. It's very straightforward. You simply seed your attached cells, incubate them to 80% confluent, and then check to make sure the cells are healthy and attached. Once you've done that, the freezing protocol is very simple. It's 3 steps really. Empty the culture media, which takes about 15 seconds. Add the freezing medium. You leave it in there not much more than 60 seconds. That's fine. Then empty that freezing media, which takes another 15 seconds, and then put it into the liquid phase in your dewar or ultra low temperature freezer. That's it. It's that simple. If you want to vitrify, you can get that with a flash freezing by following the same 3 steps. Brief exposure to the freezing media, pull off as much as you can to get as thin film as possible on the cells, and then plunge directly into liquid nitrogen. Both methods work. The key, like I said, is to get a very thin film. So you would operate the Petaka just as shown in the picture vertically. You aspirate so you can pull all the residual excess media off. You get a thin film, no more than say 20 to 30 microns. And then when you freeze, whether in the ultra low temperature freezer or in vapor phase or in the liquid nitrogen, you get the cells attached in their native configuration, frozen on the sidewall. This was our protocol here. This was our results from our experiments. 40 Petaka seeded with CHO, 50 with HeLa. We froze them away for 5 years, and every year we took samples, year 1, 2, 3, 4, 5. We were looking for attachment, viability, and then the ability to restore the culture and continue growing. The thawing procedure is very simple. It's completely closed. There's nothing open about it. You simply take 25 mils of warm culture media, inject through the sterile silicone port, and put it back in the incubator. There's no rinse because we freeze with less than 1 mil of 10% DMSO. So when you fill that to 25 mils total on thawing, you're left with less than 0.4% DMSO, so not cytotoxic. Here are pictures of our CHO samples after freezing 6 months. The picture on the left is 1 hour post-thaw, and the picture on the right is 2 hours post-thaw. I can report that we observed very high viability, 85 to 90% range for all years observed with very tight standard deviations. And the cultures were restored very rapidly. So within 24 hours, we observed pre-thaw or pre-freeze doubling times. So the cells don't have to reattach. They're already attached in their native configuration, and they rebound very, very rapidly. So healthy cells. So in this case, we were able to take advantage of a new freezing technique and a new container technology, namely the Petaka G3, where we were able to get away from cryotubes. But I think more importantly, we were able to exploit the full value of the thin film flash freezing cryo techniques out there that's been described in the literature, but to do it in a way where we eliminate all transfers, all trypsinizations. We never have to transfer thaw or unthaw in an unsanitary open way. We preserve the extracellular matrix and the cell sheet or the tissue morphology, and we did it in a hygienic way using the Petaka G3 that's closed and already in GMP cell production use around the world. So in a way, we think this is a nice advance in the way of taking advantage of these new methodologies that doesn't require any special equipment or training, and that's scalable and that's proven. So we hope you see the same value that we did. So here's the summary again. Slow freezing in CRYOVIALS has major limitations and drawbacks. It's not amenable to 2D or cell sheet preservation. And alternative methods exist, but they require these ad hoc or custom-made solutions. Our solution and protocol is very simple. It requires very little cryoprotectant, and we achieved high viability with very little cell detachment and very rapid recovery. Our method uses closed, sterile processing and provides very high cell density storage comparable to what you'd get in a 9x9 box of cryovials, somewhere on the order of 170 to 180 million cells per cryo box if you're keeping count. This method requires no special equipment or training. It can be implemented immediately in any lab, and the Petaka G3 is proven in the market, and it features a really nice cryo-resistant universal unique device identifier label, and it's commercially available and scalable. So I appreciate your time. I think we're going to move to a Q&A session after this. Thank you very much.